We know there's a lot of diversity there, but we could not really resolve those individual MGEs without using the high C approach.
Hello, hello. Hello, Irene. Hello, invisible viewers. Today I am joined by one of my friends and researchers, Irene Newton at Indiana University. We have recently published a cool paper. Well, they have published a cool paper, and I was a helpful bystander. But this paper was very interesting, so interesting that it made me purchase this hat and wear it.
Lucky for you, there's like a lot of bee paraphernalia out there.
There's going to be so much bee stuff. This is going to have the highest concentration of bee material in 10 minutes of video.
Yep.
So Irene works on something very interesting, very cool. She works on bees and also the microbiome of bees. And so we, Phase Genomics, my company, and we worked with Irene's lab to do some cool science. And we recently published, and then they published a paper about it. Irene, would you be kind enough to introduce yourself a little bit and say a little bit about your work, why it's cool beyond the obvious that it's bees, and sort of why it's important and what kind of things you guys work on?
Sure. My name is Irene Newton, and I am a professor in the Department of Biology at Indiana University Bloomington. I am the section associate chair for microbiology and also the editor-in-chief of the ASM journal MRA. So I come from a background of symbiont genomics and understanding the functions of different microbes in symbiosis with their hosts. Half of my lab works on a completely different system called Wolbachia, which is a very highly prevalent intracellular infection found in lots of different arthropods, as well as filarial nematodes. And the other half of the lab studies the charismatic honeybee microbiome from the lens of how host behavior influences the exposure that these microbes get to each other, to mobile genetic elements, to the outside world, because it's not a closed system at all, and there's a lot of interesting interactions that bees have with each other. So we were very excited to pair up with y'all to try to use the phase genomic approach to identify or link the mobile genetic elements to the little microbes that are in the bee gut, because we know there's a lot of diversity there, but we could not really resolve those individual MGEs without using the high C approach.
And we were abuzz with interest when the paper came out. But it's very cool. I mean, so the symbiosis thing is always super interesting to me because, usually when we do genomics or resequencing the genome of something, we're thinking about a particular organism. And here we're talking about organisms that live inside other organisms.
Yes.
And then part of it also we worked on was phages, which are viruses that infect bacteria. So the phages live inside the bacteria.
Yes.
The bacteria live inside the bees.
Yes.
And then the bees live
in the colony.
Yeah.
And they live on your head. They live in the colony and the superorganism.
Yeah.
So yeah, it's all connected.
So what would you say was one or two of the most interesting things about that story? Because it was very unique. It wasn't just looking at, sequencing data from bee poop. It was more.
Yeah. So there were a couple of things that we did ahead of time to make the project answer some interesting questions. One is that we used single drone inseminated colonies. So these are, I will back up and explain a little bit about bee genetics to you, but bees are haplodiploid. So queens come and females come from fertilized eggs, male come from unfertilized eggs. The queens naturally would mate multiply. So they will go on their mating flight and mate with somewhere like 10, 15 drones, keep that sperm for the rest of their life and use it to make those fertilized eggs, those worker bees. But you can artificially inseminate newly emerged queens with single drones and thereby making very uniform as you can get colonies. So where the sisters are 75% related to each other. And so we had 3 individual colonies that were single drone inseminated, and we age matched the workers so they were exactly the same age across all the colonies. And so when we sampled them, they were as controlled as you could be. So really allowing us to ask whether there was an influence of the colony and genetic background, in addition to this broader question of what mobile genetic elements are there and how they might be linked to the microbial community.
That's super cool. And so why do we want to know what mobile genetic elements are?
Just because I want to know, Ivan. All right, I just want to know.
So I'll tell you, I'll spike you the answer. So when we first looked at the data, right? So one of the things that, for those of you who are watching who maybe don't know, so one of the unique things that Phases Tech does in microbiome is that you can look at microbiome sample and you can tell where all the phages and all the plasmids are coming from because it connects them to the bacteria. Normally you don't know, you sequence the DNA, they're just all over the place, but our tech will actually connect them. So you're looking at the microbiome, you're identifying the bacteria, you're also identifying where the plasmids are. And plasmids, of course, carry, antibiotic resistance.
Yes, they do.
That's one of their big things. So one of the reasons why antibiotic resistance flares up so quickly across the world is that it travels on plasmids, which are these little circles of DNA that jump around from bacteria to bacteria.
Yep.
And when I first looked at the data that came out from your stuff, I was super surprised because there was antibiotic resistance all over the place.
Yes.
Now, we've done a lot of work in like cows, and people who are undergoing antibiotic treatment and other places. And so I'm used to seeing but normally if you sequence the poop from like a worm or something, you're not going to see all that antibiotic resistance.
Right. So where did it come from, right? Where did it come from? And that's because the bees have like a history of tetracycline treatment. So it's not allowed anymore, but it used to be kind of a thing that beekeepers would do prophylactically, treating their bees with antibiotics, tetracycline being the most common one recently. So the honeybee microbiome, previous publications of just like isolate sequencing and then a few metagenomic analyses identified that there were tetracycline resistance genes in the DNA you'd isolate from a bee. But what was really cool in this case is that you could link that those tetracycline resistance cassettes are actually on plasmids and that those plasmids have very broad host ranges. So you could actually show the core community of the bee is sharing these plasmids with each other.
So question, because I don't know the space very much, do bees from colony A ever go to visit colony B? Is it possible that they'll spread it between different colonies?
Yes. So especially around now, this time of year where maybe the last of the goldenrod is out and we're running out of resources, we're entering this dearth, and the bees are
November 3rd at recording.
That's right. And so the bees are freaking out, like they're running out of floral resources, and they will rob neighboring colonies that are weak. So there are bees that serve to guard the entrance and prevent these intruders from coming in. But if a bee colony is not doing very well, then a stronger colony can overwhelm it and can pillage all of their forage resources effectively. So they can enter each other's colonies for sure.
So is it possible that I guess I have two questions. One is, can they transmit some of these mobile elements to other bees? Can they give each other, basically spread antibiotic resistance?
Yeah.
And can they actually, are they vectors for AMR in general? Are they flying around and just spreading it everywhere?
This is a great question. And so as of now, right, based on our data, we have this snapshot. The snapshot says, we know these cassettes are encoded on mobilizable elements and that those elements within a colony are shared. And in those 3 distinct colonies we sequence, the plasmid backbones share relaxases. So we know that they're similar across these colonies. They're not identical, but they are similar. So it is possible that microbes and or their MGEs could be spread that way. It's also possible that they could spread them on flowers. We know flowers are vectors for bee diseases overall. So they've been observed for, pollinators kind of are spreading the STIs of plants, if you will, right? That's what they're doing. So they know they spread plant diseases that way as well. And so we're really curious now. We have a time series data set that we're analyzing now to try to really get at the dynamics of these MGEs. So what exactly does happen to these plasmids over the winter or as across colonies or if you have a colony in the same apiary, do the colonies retain some colony-specific MGE signature over time, or are these really dynamic and we just don't see them ever again? Like, what's the churn like in these mobile genetic elements?
Yeah. Well, it's really that's super interesting, and a lot of it, like I keep thinking about. I think it's less in the news now, but it used to be like the bee sort of like the hive collapse, colony collapse disease, and all this stuff. Right, there was a disease going around, and there was also linked with pesticides.
Yes,
but I don't hear about it much anymore, and I've heard some sort of positive news about bees somewhere. I forget where. What's the deal?
What's the news? What's the news on bees? Yeah, so I think
bee status report, please.
I say people always ask, how are the bees doing? Right? How are the bees doing? So I always say, like, are you talking about our research bees? Or are you talking about bees overall?
Hardworking science bees with little lab coats and a tiny pipetter.
Exactly. But yeah, I think there was, back in 2010, I think, was the peak of this colony collapse disorder phenomenon that has been linked to the use of pesticides. At least I would say to how the exposure to pesticides can alter the ability of the bee to orient, can alter your ability to navigate, so that the specific syndrome that was observed was that workers would leave and then they would not come back. So you never found like a pile of dead bees or anything like you would with normal pesticide poisoning. Now that's to say, like, this is a very specific kind of syndrome that was observed, but bee numbers had been in decline. The number of colonies that beekeepers can maintain had been in decline for a long time, and as had longevity of queens, so the number of years that a queen would live. So that has plateaued some in the last few surveys. But I think maybe, not sure if it's exactly the positive news you heard, but people have been focusing a lot on nutrition as a primary driver of all of these. So as floral landscapes really changed, as monoculture crops became dominant in North America, pollinators writ large just really had access to less nutritious floral resources. There's fewer of them and less diversity. And the bees really need a really large, broad source of flowers to visit, of other nectaries. And so if they don't have that, then it becomes more difficult to sustain them. But if you can supplement them with specific nutrients, and either they be fungal or they be plant derived, then they can survive better for sure.
Cool. So then, so you and your team that published this paper, right? First author is named Chris. Maybe he will appear here in the video later.
Chris Robinson is a grad student in my research.
It's possible.
Yeah.
And what are you guys doing next? What's next in the world of bee microbiome?
Yeah, I've become as part of this larger institute called, so NSF previously, before all the chaos, funded these biology integration institutes that their goal was to try and integrate within and between the sciences. So within biology, to try to break down silos between molecular and organismal or evolutionary science, for example, and then bridge to computer science or to chemistry, et cetera. And that institute had as its goal try to understand how mobile genetic elements alter microbial behavior and the effects this might have on ecology, especially in host-associated systems. And so I'm interested in that, but I'm also interested in how the behavior of the bee and the fact that they go and visit all these flowers and interact with each other influences the MGEs that these organisms are exposed to. And then that has an effect on their physiology. So there's like feedback loops, up and down this chain. So we have projects in the lab manipulating the behavioral environment of bees and looking at how these MGEs are transmitted. And we have projects in the lab looking at how the MGEs directly influence the behavior of bacteria with regards to important traits that we know are like needed for host association. And we have projects in the lab like Chris's time series coming up, trying to look at the dynamics of these MGEs. One of my favorite hypotheses for this huge zoo of plasmids and phages we discovered as part of this Phase Genomics project is like, wow, like how can there be so many thousands, right, of these MGEs? And my favorite hypothesis is that it's all garbage. But like the fodder of evolution, right? Like you need to have all this diversity there to have something to select for. You need to have just like this zoo that you see when you take the snapshot. But then over time, presumably, if something is positively selected in that environment, we will see a signature of its presence increase. So that's the stuff I'm excited about, is just how this process of going to visit nectaries and bringing all that stuff back to the colony brings MGEs with it, and how that then impacts the evolution of the microbial community that's associated.
And that's super interesting, because this idea of having like a diverse microbial sort of smorgasbord has been kind of pretty common in the microbiome space. I mean, that's our running hypothesis for why FMTs work. But to think of that same idea, except for instead of the diversity of microbes versus the plasmids and the phages, like the mobile elements, that there's also this kind of huge pool of them that's constantly being spread around and moved, and that actually forms. It's actually extremely important for biology. We just have never really been able to see it before.
Exactly, we haven't been able to actually dissect it and actually figure out, who they're associated with and what are they carrying.
Yeah.
And it's so hard to do those experiments in vivo, right? Like we can do lots of experiments in the lab, right? We've got phage hunters going out to try to get these individual phages, but that's what we find is so different from what you get from sequencing the whole community, right? We're selecting in the lab for something very different and possibly really rare, like this completely lytic phage that infects this particular microbe super well. Right. So I think it's a much more of a gradient of association in natural communities with a lot more diversity that's present. And so that's part of what we're capturing with the sequencing. Yeah.
No, it's super cool. I mean, we've seen the same thing, that when you grow stuff up, it's completely different than what's actually there if you capture the community itself. So, I mean, that's super exciting. Irene, thanks for taking the time.
Yeah,
I had a great time. That was a cute thing. I'm going to ask you, what is your lab, science lab guilty pleasure? Like some people will be like, I like to use extra parafilm or whatever, or or that kind of thing.
I'll tell you like an obnoxious thing I do.
What is your most obnoxious lab habit?
My lab guilty pleasure. So I do some wet bench science, and I'm also a computational biologist. And sometimes when I'm trying to run stuff in the computing cluster, I feel like my job is just not being prioritized. So my horrible thing that I do is I go on the cluster and I look at the queue, and then I look to see who those users are. And then I email them.
Oh, I thought you were going to say I run down there with a sack full of nickels.
I email the HPC people, and I'm like, I don't understand why my job is waiting, but all these other people are getting priority.
That's an entrepreneurial spirit. That's
is
what
it?
you got to do. You just go like,
it's a bratty thing to do, I think.
I'm just gonna do this. Yes. Well, that's very fun. I'm sure you have lots of friends in the
Exactly, making friends left and right.
Yeah. Cool. Well, Irene, thanks again.
You're welcome.
We'll keep in touch and publish many more papers about bees or Wolbachia or other things. For the rest of you who are listening,
you can link to the paper.
That's right. Check out the paper. Smash that subscribe button if there is one. And and thank you for watching my conversation with the queen bee herself, Irene. Thanks.